Recording. I'm going to start in 3 minutes. Okay, I'm going to take an image for attendance. A 1 and a 2 and a 3. Yeah. One more image from the second sheet. I'm glad some of y'all are outside enjoying the fresh air. Okay, today half of y'all are going to come to lab. It would be a good day to wear your tennis shoes rather than something like fancy church shoes because we may be doing some exercise. All right, it's 10:30, so this is November 16th. We're going to continue to talk about renal function today. I don't see any chats or any hands being raised for any announcements. If you got something, you can let me know about 10 minutes before we end. Okay. Last time we were together, we went over sort of an introduction to the kidney and the kidney tubules and the functional unit of the kidney. That functional unit is the nephron. And it would be a good idea to practice drawing the nephron and staring at that image in the textbook. It's usually pretty easy to get images of nephrons. They're all over different anatomy and physiology textbooks. I would like for you to be able to talk about where salts are actively pumped, where water leaves the lumen of the tubules, where urea is having an effect, right? So I'm going to go through some of those steps again right now. I'm going to start with the afferent arteriole today, coming in over here, comes into a capillary network that breaks up into many capillaries. Right now, I'm just talking about the blood stuff. And it leaves the renal corpuscle through the efferent arteriole. So this is an arteriole bringing blood in. This is the arteriole bringing blood out, right? And this is kind of like at the core of the nephron. It's the core of the renal corpuscle. On the outside here, catching something that's coming out of the blood is Bowman's capsule. So this whole big circle here is called renal corpuscle. In the very middle is the glomerulus. On the outside is Bowman's capsule. Bowman's capsule is catching this fluid, right? While the blood comes in, red blood cells stay in here, big proteins stay in here, and they keep going. But salts, wastes, and water come out. Right. Then we have the proximal tubule. It's proximal because it's close to the renal corpuscle. I'm drawing this as a tube here, but I want you in your mind's eye to know that these tubes are definitely made of cells. They're an epithelium, right? So I could draw my cells here. A good way to remind a reader that these are cells. You put a little nucleus in each one of them. Okay. One thing that's important here, means you kind of understand it, is to understand the difference between the basolateral side of these cells and the apical side of these cells. Like the apical side is sort of the front of the cells. The basolateral side is sort of like the back of the cells. So if I was drawing this cell a little bit bigger, this is how I would draw it. Back here is the basolateral membrane of the cell. There's cells on this side too, right? What's important about that? It's because back here, away from the lumen, this is where those sodium, potassium, ATPases are. They're working back here. That's important because they are going to be pushing sodium out, 3 sodiums, while 2 potassiums go inside here, right? And this is some of the osmotic force that allows for aquaporins to be working here, right? So I'm in one cell here. You can imagine some water channels that if there's more sodium on this side, that's going to help water be pulled through this cell out of the lumen into the interstitium. And once it's back here in the interstitium, it's available to be picked up by blood. All right. So this is the lumen on this side. And what's this juice right here? That fluid is called the ultrafiltrate. It's not called urine yet, even though sometimes I sort of slip up and I might describe this as urine, because what this ultrafiltrate is more or less like pre-urine. It's the precursor to urine. And we have to concentrate it, more or less take the water back out of it before we call it urine. Right, so here's the proximal tubule. Continues all the way down, makes a loop. It makes a loop kind of deep in the renal pyramid. I'm going to draw it short just so you can see it on the board here. Right. Now, last time, so that you could understand what I was drawing, I draw the simple version of the nephron, which goes like this. You can see the proximal tubule, you can see the loop of Henle, and you can see the distal tubule. And I'll even finish it right here just so you can remember what I'm talking about. Right? I'm going to erase this in a little bit, but the simple way to do this to show where cells are allowing for the movement of water is to put proximal tubule, loop of Henle, distal tubule into the collecting duct like this. Okay, so what has happened right here is this distal tubule is draining into the collecting duct. Now this collecting duct, it's getting distal tubules from several other nephrons. So whenever you're like defining the nephron, really the nephron starts here at this renal corpuscle, goes through proximal, loop of Henle, distal tubule, and kind of by the books, the nephron ends there. And many nephrons hook into that collecting duct. Okay, I want you to know about that, but today I'm going to draw this in a slightly more accurate way. And it's because really the distal tubule doesn't just go into the collecting duct. It comes back around the back, comes very close to this glomerulus here, right? If you look closely for it in most textbooks, they also show this more detailed drawing. Really, this distal tubule comes back around. I am still the distal tubule here. It comes back around and it comes right here close to the glomerulus and the afferent arteriole before it finally loops around into the collecting duct. All right. I don't know if you can see it, but what's important is to not think that the distal tubule, it's not emptying into this Bowman's capsule or anything. I'd like for you to think of it as going around the back. It's going around the back. It's not allowing ultrafiltrate to leak into here, but it is close here on purpose because there are some cells that fill up this interstitium area. Let's say if this is the afferent arteriole, there's these in-between cells. And they're really kind of in the interstitium. They're not really right here. They're not part of this distal tubule. And the ones that I'm drawing here are not part of the afferent arteriole, right? What I'm drawing here is what we call the juxtaglomerular apparatus. Okay, so as an abbreviation, it's the JGA. This is how you spell that. Juxta. Juxta means close, like juxtaposed. Juxta glomerular apparatus. It's called an apparatus. That's a pretty nerdy term because I think of an apparatus as a little machine. You could think of it as juxtaglomerular monitoring system or something like that, because what we're drawing here is the quality control monitoring system that makes sure that we don't lose too much urine. Okay. What we're going to talk about here is how this nephron conserves water. In other words, we're going to talk about how this nephron stops us from becoming dehydrated. That's what we're going to talk about. Okay. So you got these cells in the in-between area, and these cells are called the juxtaglomerular cells. Juxtaglomerular cells are around the outside. There's another group of cells that are on the outside of this afferent arteriole. They are modified smooth muscle cells. So I'm going to put them right here, right around the cells of the capillary. Granular cells. Called granular because when you look at them in the microscope, they really do seem like they have a lot of stuff in their cytoplasm, right? I'm going to give you another group of cells. group of cells that are part of this distal tubule. So watch my finger here. I'm on the distal tubule. I'm on the distal tubule here, and this is the continued distal tubule. I'm going to give it a D right here. The cells that line the distal tubule right here, they have a special name. You could say they're continuing right here as well. Megan Boulanger, you know the name of these cells? These cells that are lining the distal tubule, they're specialized distal tubule cells, but they're coming up right behind the glomerulus.
No, but I can guess endothelial.
That's a good guess. Annalise, you ever heard of these?
No, sir.
Okay. These have a special name. These cells right here are called the macula densa cells. Macula densa. You look at them under the scope and they look very dense. A macula is kind of like a term like for a body or something, right? So I'm gonna draw that out. Macula densa. Macula densa cells. They're lining that distal tubule. All right. I'm going to draw some other organs that take part in this story. One organ, you can probably recognize it from the way that I draw it. You know, Miss Julia, you know what that's looking like? Miss Southall?
The hypothalamus?
Yeah, and the pituitary right underneath it, right? The anterior and the posterior pituitary. I'm going to draw another organ that sort of helps out here. That's the liver. All right. I'm going to draw some lungs. They're not anatomically correct, but they all have a role here. All right, let's think about what's going on in the urine, the ultrafiltrate, and the blood when you're about to be dehydrated. One way that that happens is if your ultrafiltrate has too much water in it, right? Your urine is about to be too watery. You're about to lose too much urine. You're about to be dehydrated. That's what this nephron is looking at every day a little bit. Okay. But our nephrons, because we're healthy, it doesn't let us lose too much urine. So how does it know? It's because these cells of the macula densa, you can consider them as being monitoring cells and being able to respond to the ultrafiltrate as being either too salty or too watery, right? We all have this, you can think of it as tasting, because I think it's pretty easy to think about water with no salt. You can practically taste that, or a little bit of salt water like at Grand Isle. You go swimming at Grand Isle, you can taste, oh yeah, this is salty water, right? A margarita is salty, but a bourbon and Coke is not, right? So these cells can tell the difference between salty ultrafiltrate or watery ultrafiltrate. If the ultrafiltrate is too watery, these macula densa cells, they send out the alarm. Courtney, you ever heard this story before? We're going to talk about the alarm here. Miss Bertini, you know what happens?
No.
All right. I'm pointing to the macula densa cells here. They have just tasted that this ultrafiltrate's too watery. They communicate with these cells right here. These are the juxtaglomerular cells. The juxtaglomerular cells communicate with the cells right next to them, the granular cells, and a cascade is kicked off. This cascade is called the renin angiotensin system. Okay, you could say that what we're learning today is how we concentrate urine, but you could also think today that what we're going over is the renin angiotensin cascade. Angiotensin. Okay, this is how it starts. These cells here that are lining the afferent arteriole, these smooth muscle cells, they start this off by releasing renin. Now, renin is a protease. Everybody knows what a protease does. A protease cleaves other proteins, right? There's the liver here. The liver releases a precursor molecule called angiotensinogen. Angiotensinogen. Right? You might recognize this terminology. It's like pepsinogen was a precursor. Angiotensinogen is another precursor. It comes into contact with renin. You know what renin does? Renin cleaves other proteins. So it cleaves angiotensinogen into angiotensin. I'm going to call it AGT1. Angiotensin 1 is pretty active, but it's not as active as it could be. Angiotensin 1 goes to the lungs, and the lungs release another cleaving molecule, and that's called ACE, angiotensin converting enzyme. If you've ever done a PowerPoint about high blood pressure, angiotensin converting enzyme is important for this. So we'll talk about that in a little bit, but angiotensin converting enzyme made by the lungs has an effect on angiotensin 1, and it cleaves it into angiotensin 2. Now, angiotensin 2 is active. It's the main active hormone here, right? So this is a cell signal that's being sent throughout the blood, and it has two big effects. One effect is not really on the kidney that I'm going to draw right here, but on this gland that's on top of the kidney. That's where the adrenal gland is. When angiotensin 2 comes to the kidney, it responds, the adrenal gland responds by releasing aldosterone. Aldosterone. That's the first thing angiotensin does. Another thing that angiotensin does, angiotensin 2, comes up to the posterior pituitary, and it leads the posterior pituitary to secreting what's called ADH, antidiuretic hormone. Some books call it vasopressin, but I'd like for you to think of it as antidiuretic hormone. All right, 2 cell signals now have been released. I'd like for you to think of these as like the 1-2 punch. They were both released because of angiotensin 2. I'm going to put a 1 next to this. It's part of the 1-2 punch. I'm going to put a 2 next to this one, right? Now, all of this started at the distal tubule, and these 2 molecules, they kind of come back to the same area, and they create a change here, right? What change do we need? What these macula densa cells recognize was that there's too much water in this ultrafiltrate. So what do we need to do? We need to reabsorb more water. So that starts by aldosterone. Aldosterone goes throughout the blood, and it comes to this distal tubule, and it says to the cells on this distal tubule, y'all need to start using your sodium potassium ATPase. It's on the back of these cells, right? And they start pumping sodium out. So they're pulling sodium out of the ultrafiltrate. And you probably know what's going to happen next, but we need aquaporins. And that's the job of the antidiuretic hormone. Antidiuretic hormone comes in, joins this party. It especially has an effect on the collecting ducts, but kind of in the same interstitium, right? The collecting duct is also made up of cells. And what antidiuretic hormone is doing is it's opening up aquaporins here. All right. So what do we get happening here? We get sodium being pulled out of the ultrafiltrate by the sodium potassium ATPase. So there's more sodium out here, more sodium. And we have aquaporins that have been opened and brought to the membrane here. And so now water moves into this interstitium, water. Some water might be being pulled out of this ultrafiltrate in the distal tubule too. So we have fixed the situation, right? What this whole system did, this angiotensin, this renin-angiotensin system, is a cascade that leads to the distal tubule turning on to move water out of the ultrafiltrate back into the interstitium. It's a way to reabsorb water. By taking the water out of the ultrafiltrate, now the urine that leaves this collecting duct, instead of having a lot of water, it only has a little water in it. Okay, so what we have done is we just conserved water. Right. It doesn't feel like you're having to think about that every day, all day, but in a normal student or faculty, you only let out about 1 liter of urine every day. If this breaks down, you would lose more. So this system makes sure you only lose about 1 liter of urine. It's a way to keep you from being dehydrated. Okay, Cameron, I'd like for you to think about these two extremes when you think about how the kidney works. One is that we always fight dehydration. But Cameron, do you remember the other extreme that you have to fight with the kidney? You still muted?
Blood pressure?
High blood pressure, right? That's on the other end. Once you start getting older, you start having trouble with high blood pressure. What's a way to think about that? It's the opposite of what we were just talking about. In high blood pressure, it's almost like saying your blood has too much water in it, too much volume. Okay. So what do we need to do? When you have high blood pressure, the way that our homeostatic mechanisms fix that is they allow for more urination. If you make more urination, then that relieves high blood pressure a little bit. All right, I'm going to show you how that happens. Okay. In a normal situation, this renin-angiotensin system is working. I'm going to put another organ up here. That other organ is an organ that we talked about kind of a long time ago. That's the heart. Now, the part of the heart that gets venous return, it's sort of a place to monitor how much blood is in the whole system, is the right atrium. And when the right atrium is stretched out because there's too much blood in the system, otherwise, another way to say that is there's too much blood pressure, it's high blood pressure. When this right atrium is stretched out, it responds by secreting ANP. ANP stands for atrial. It makes sense because it's coming from the atria. Atrial natriuretic peptide. Natriuretic peptide. Atrial natriuretic peptide. When you have a term that says uretic, that's a hint that what it's going to do is going to lead to more urination. Atrial natriuretic peptide comes up to the hypothalamus. A while ago, the hypothalamus was allowing for ADH to be released from the posterior pituitary, but ANP inhibits ADH. Another way that we show that in cartoons is instead of an arrow, you kind of put a T right here, right? So the heart releases ANP, which inhibits ADH. With ADH inhibited, we might have sodium being pumped here, but what we don't have is we don't have water moving, because even though the cells are here, their aquaporins are closed. So when we turn off ADH, it closes these aquaporins and does not allow water to be taken from the collecting duct into the interstitium. So what does that allow? That allows for more urine. Okay. You could call it diuresis. It causes more urination, or in medical terms, that's diuresis. All right. Those are two normal ways that the kidney responds to these two extremes. And really every day, all day, the kidney is not responding to an extreme situation, but instead it's sort of keeping just a light balance between these. So the kidney is helping to conserve water, and the kidney is also helping to inhibit high blood pressure, right? Because it's not really creating a big old pulse of renin. It's just creating a little bit of renin, okay? It's not creating a big old pulse of atrial natriuretic peptide. It's just creating a little bit of that. So that helps turn off ADH, and the renin-angiotensin system allows you to turn on ADH. All right. So in this situation, these were normal responses. A while ago, we made less urine to conserve water. Right now, we're making more urine to help us with high blood pressure. But sometimes this can go wrong in a healthy individual that does not have high blood pressure. All right? What can happen? Like, let's say you just got finished classes on Wednesday, next Wednesday. You're going on Thanksgiving break, our big 2-day break, right? What can happen is maybe you make a pass by Roxas if it's open, and you have a martini. You know, a martini only has a little bit of juice in it. You drink that down, you start to feel good, so you have another martini. Right? It's still not a lot of juice, but you drink that, and all of a sudden you got to use a restroom, right? And then before you head home to see your folks, because Thanksgiving holiday is coming up, you have a third martini, and now it's happened. You broke the seal, and you start peeing a lot. All right. If you've drank alcohol or maybe coffee, you kind of know that feeling. It's like, man, I'm peeing a lot. That feeling, that weird feeling, is because alcohol also inhibits ADH, right? But what's tricky is that you don't have high blood pressure. So you don't really want to inhibit ADH. You are normal. So in a normal situation, when you have alcohol, we could call it ROH. That's like ethanol. We'll call it ethanol if we want to. It can be beer or martini or moonshine or wine. Ethanol goes and turns off ADH. So the end effect is the same thing we were just talking about with ANP. Atrial natriuretic peptide turns off ADH. Alcohol also turns off ADH. What happens when you turn off ADH? you start making more urine. In the case that I'm talking about right now, this is an artificial release of more urine because you don't want to release urine. It's an abnormal release of urine. So you pee more and more, right? You become dehydrated. And you might not feel it until the next morning. And the next morning you get hangover. Okay. So some of us have felt that. What hangover is dehydration. And it almost is counterintuitive because you feel like it's happening when you're drinking. But if you were drinking just water, you wouldn't turn off your ADH. So it happens when you drink alcohol. Alcohol turns off your ADH, leads to more urination, which actually allows you to lose too much water. It's kind of like this monitoring system has been turned off. Leads to dehydration and hangover. And that's why practically all you usually need to do for a hangover is maybe drink some Gatorade or some water or something like that and rehydrate. Usually gets better. Or you can ignore it and have another beer. Okay. So those are three situations that I'd like for you to be able to think about with the nephron and the quality control of the nephron. That the first one is how the nephron helps us conserve water, how the nephron helps us fight high blood pressure, and how the nephron can be abnormally influenced by alcohol to actually cause you to go through dehydration. All right. Now, I'm going to sort of review this before I totally erase it about how this nephron works. Make sure you've heard it all in one go. Okay. You know what's right here? That's the renal corpuscle. Blood comes in, blood goes out. As this blood goes through the glomerulus, salt and water pass through. That's the ultrafiltrate. But I want to make sure that you know that's going on right here at this proximal tubule is there's sodium, potassium, ATPases on the backside of this epithelium that's making up these proximal tubules, and they're automatically always pushing out salt. And there's open aquaporins here that are automatically always allowing water to follow the salt. Okay. So there's not much decision being made here. As that ultrafiltrate comes through the proximal tubules, sodium, potassium, ATPase is running, pushing salt out here into the interstitium. And these aquaporins, they're automatically here and they're already open. And so a lot of times that these textbooks will say almost 75% of the water in the ultrafiltrate is pulled out of the proximal tubule. So a lot of the reabsorption happens right here automatically. Okay. Then what happens when the ultrafiltrate gets down here in the loop of Henle? One part of the dogma is that there's not much sodium, potassium, ATPase working down here, but instead this ultrafiltrate can be concentrated by there being another osmotic particle out here, and that's urea. Urea can be called an organic osmolyte. It's an osmolyte. It's an osmotic particle like a salt, but it's not a salt. It's not sodium or chloride. It's organic. It's got a nitrogenous molecule in it, right? And urea is so high down here at the bottom of the renal pyramid that it also pulls water. And it's pulling water from this collecting tubule too. Okay, so the urea down here at the bottom of the renal pyramid is here acting, giving osmotic pressure and pulling water. It's another way to take back water. And so that's why these two sections, the proximal tubule and the loop of Henle, they kind of automatically take back a lot of water. And right here, this monitoring system is making that decision. okay, did we pull too much water out already? Or did we not pull enough water out? So that's why the distal tubule, it's like the last chance to make the decision. Do I turn on the sodium potassium ATPases to pull out more water, or do I let it go out the way it is? Macula densa is tasting that ultrafiltrate. Is it too salty or is it too watery? If it's too watery, you're about to lose too much urine. If it's too salty, then your blood might have too much water in it or high blood pressure. All right. Okay, so that's going on here. What we just went over was how we compensate, how the macula densa either turns on and leads to us absorbing more water out of the ultrafiltrate, or it doesn't turn on to allow more water to go out. Now, the last part of the puzzle that I haven't drawn for you, it tends to be kind of complicates the picture, but I'm going to draw it for you now, is what happens downstream of this efferent arteriole, right? This is the afferent arteriole. The blood is coming in here from the heart. The blood goes through the glomerulus and it loses ultrafiltrate. But then some blood is coming out here through the efferent arteriole. Okay, this blood, it doesn't just go back to the heart. This blood wraps around the renal tubules. This is the efferent arteriole here. And this blood comes as vessels and it wraps all around this renal nephron before heading back to the heart. So this vasculature after the efferent arteriole, it has a special name. It's called the vasa recta. The vasa recta. Recta kind of means like it rectifies the situation. Vasa has to do with vasculature, so that makes sense. This vasa recta has this important role of, maybe you can see how it's going to work here, because this blood is coming into close contact with all that sodium that was here and all that water that's being pulled to the sodium, right? So when I told you that the water was coming out of the collecting duct into the interstitium, it's not just sitting here in the interstitium. The water that's coming out of the collecting duct, it goes into this vasa recta, and the blood takes back the water, right? As water is taken out of the distal tubule, it goes back into this vasa recta. So now that reabsorbed water that left the ultrafiltrate is filling up this vasa recta. And so that's why this blood that comes in and the blood that finally leaves is pretty similar, except it just left a little bit of salt and water and waste that became the urine, just a little bit. Now this vasa recta brings the blood back to the heart without some of those wastes. All right. Y'all got some questions? Did I go over that too much, too fast, too little? Okay. That's how it happens in the human kidney. That's how we concentrate our urine. That's how we do these two important steps. That is filtration right here at the renal corpuscle and reabsorption in the tubules and the vasa recta. Reabsorption of that water. Right. I'm going to talk about some other things. What we've been going over is, I want to make sure you understand how this works in us as mammals. Now, humans are kind of middle of the road mammals. We're not in the desert, right? We're pretty good thinkers, but like the way our kidney works is not necessarily some extreme, crazy kidney function, because there are some animals that are at the extremes, like there's little mice that live in the desert. What you find in mammals that are in the desert is they have a real long loop of Henle. So mammalian biologists recognize that having a long loop of Henle is directly correlated to whether you can concentrate your urine or not. Or another way to say that is, if you have a long loop of Henle, you can conserve water better. The loop of Henle seems to be sort of a new thing in animals, like mammals have it and birds have it, but other animals don't in our group, like snakes and turtles. They don't have a loop of Henle. Frogs and fish, they don't have a loop of Henle. It's sort of a new structure that you find in birds and mammals. Okay. So one direction I would like to go back to right now is I'd like for us to think about fish again. Okay. The first way that I described the fish was that fish are our cousins, you know, and yet we have fish that are at these two different extremes of conserving water. We have some fish in salt water, some fish in fresh water. It makes it even easier to kind of compare this when you have a family of fish so that these fish even look alike. Our example of that was the Gaspa goo. You could just call it the goo fish, the goo versus an Atlantic croaker. Maybe you could say the black drum. These are in the same family, cyanids. But they're having to do two very different things, right? What are they fighting? This fish in salt water, he is exposed to more osmotic pressure on the outside of his body. There's more salt out here. So he's always in danger of losing water. Water wants to be pulled out of his body, right? A fish that's in salt water hardly makes any urine. They have to conserve water. They can't really drink the water. Oftentimes it's thought that the way they get water in their body is by eating another fish or a little shrimp, and the water that's in that shrimp's body goes through its gut into its blood. So they get water by eating. On the other hand, at their gills, they have so much salt out here, they have to pump out salt. Okay. So at their gills, the way that this is often described is that these fish secrete salt. Right. So they make no urine. They secrete salt out of their gills. On the other hand, in a freshwater fish, he's surrounded by a whole bunch of water, and in an opposite way, the salt in his blood makes him have more osmotic pressure than the surrounding water, right? You could say that this fish is hyperosmotic to its surroundings. So what's he in danger of? He's in danger of water moving into his body. The water from this low osmotic pressure fluid wants to go to where there's more salt. So this fish makes a lot of urine. On the other hand, instead of secreting salt, at his gills, he's able to find very rare salt molecules in this fresh water. This fresh water is not like deionized water, but the metals and the ions are rare, right? So they have gills that allow them to pump salt into their body. And the way that this works is not the sodium, potassium, ATPase that much here. This is done by another pump. It's called the proton pump that uses the movement of a hydrogen ion, which is the same as a proton, right? So the proton pump is on. Over here in these saltwater fish, they tend to use the sodium, potassium, ATPase. They use its activity to secrete salt out of their gills, right? So these fish are doing these pretty opposite things, even though their body is about the same size or from the same family. But what I wanted to introduce you to is, I'm talking about these fish as the goo is a fish that always lives in a lake, never goes to the salt water. Black drum is mostly a saltwater fish that you would not find in Lake Verret or Toledo Bend. You would never find that. But you can probably think of some fish that make their living going back and forth, right? These fish, they stay in their own neighborhood, but there are fish, just like there's crabs. We think of blue crab as a saltwater animal, but you can catch blue crabs in Lake Verret sometimes or in Lake des Allemands. So there are animals in an estuary that know how to osmoregulate from saltwater to freshwater. They have homeostatic mechanisms that compensate for the change from saltwater to freshwater, right? In fish, you could maybe think of some of our local fish. Like I just said, a blue crab will do that. The male blue crabs, they'll go up bayou from Fouchon, up the bayou, up to Thibodaux. That's why you see crab traps in Bayou Lafourche, even though Bayou Lafourche is very fresh right here in Thibodaux. There's crabs up there. A fish that'll kind of do that is the redfish. The mullet can do that. When you see mullet jumping in freshwater, they came from saltwater, most likely. Redfish, you imagine them hanging out around Grand Isle, but they go up Barataria Bay, up into Lake des Allemands, which is mostly freshwater. Lake des Allemands is fresh enough to be surrounded by cypress trees, living good cypress trees. So that means it's pretty fresh water. Okay. So that means these fish are having to kind of remodel their gills and remodel their kidney every time they do that. Well, a good example, a good animal model that's been used to study that is a salmon. Now, the salmon do it in a way that's not the same as like the redfish that I just mentioned. Redfish kind of go back and forth at will, but salmon mostly do this once. The salmon go upriver to spawn. And a lot of salmon that do that on the West Coast, they go upriver and spawn and then die. All right. So this is an important time in their lives. So this activity of migrating, running out of black here. Migrating upriver to spawn is called anadromy. So these are anadromous fish. An anadromous fish goes upriver to spawn, but there are fish that are opposite of that. Those are catadromous. A catadromous fish lives most of its life in fresh water and then goes to salt water to spawn. And an example of this is the eel, the American eel. They go spawn in the Sargasso Sea. Michael Claymont, you know where the Sargasso Sea is? You got to unmute.
No, sir.
The Sargasso Sea is not really like the Gulf of Mexico. It doesn't really have walls, but if you look on a map, the Sargasso Sea is pretty much where we would put the Bermuda Triangle. So the Sargasso Sea is kind of like between Bermuda, North Carolina, and the Bahamas. It's just part of the Atlantic Ocean where eels can go all the way from the Great Lakes, all the way from the upper regions of the Mississippi, all the way out. They go over there and they spawn. Okay, that's a catadromous fish. So for our example, I would like for y'all to know what an anadromous fish has to do. Okay. So I kind of told you the story here, what a salmon does. A salmon goes upriver. I'm gonna put the upriver over here. He's upriver. And the salmon lay their eggs upriver and the salmon dies. So these eggs hatch and what you get is some little baby fish. And these baby fish live in the fresh water for a little while until they begin to look a little more like a little salmon. Salmon has a little adipose tail like that. Okay. And as juveniles, these salmon finally begin to go to the sea. Right. So you could imagine this happening on the West Coast. As these fish start getting closer and closer to the sea, these fish start to become exposed to salts in the external environment. They're tasting a little bit of the salt water when they get close there, right? What these fish are using when they're up in fresh water is they're using the proton pump. That's at their gills. They're making a lot of urine. When you make a lot of urine, you even have glomeruli that are real big. Those big glomeruli allow you to make a lot of urine because you can make a lot of filtrate. As they get closer and closer to the salt, you have an endocrine difference. Okay, one difference is that cortisol is released. Cortisol is sometimes considered a stress hormone. It's released from the adrenal cortex, and growth hormone is released. Growth hormone comes from the anterior pituitary. Both of these have an effect on the gills. And if you take images of the gills, you see the cells of the gills begin to change. And you start to get a type of cell that's called a chloride cell. And these chloride cells have a basolateral membrane that has a basolateral membrane that is chock full of sodium potassium ATPase. All right. So as they get closer and closer to this salt water, cortisol and growth hormone lead to a change in their gills. Sodium potassium ATPase starts to get more active. And as they get to the salt water, boom, they change. Now that sodium potassium ATPase is allowing them to secrete salt. They stop making urine. And now they have to really conserve water. And they're in seawater. Now, salmon might live over there in seawater for 5 years. They live their adult life in seawater and they're eating and having a good time. Around 5 years old, they get to a certain size where now it's time to spawn. Okay, they have kind of like a change in their life. They're in seawater, they have to go back to fresh water. So what does that look like? An anadromous fish that's going to spawn in fresh water has got to turn all of this stuff off and turn something else on. And what you see in these fish that are going upriver, they go from a salty water to a water that's a little more fresh. One of the things that's released is prolactin. All right, Miss Henry, does that seem weird to you that a fish would release prolactin?
Yes.
Why is it weird?
Because doesn't prolactin form milk whenever fish typically do yolk and eggs?
Well, the first part of that sentence is what I was going for.
Okay.
Because fish don't make milk. Fish don't have mammary glands. It's because prolactin got named by scientists that were studying mammals. And it's true that prolactin leads to milk being made by mammary glands. But then as scientists kept looking in other vertebrates, they found prolactin in snakes, which don't make milk, frogs, which don't make milk, fish that don't make milk. And they realized, oh, prolactin has been around for a long, long time, and it has this older, more ancient function. And the function is in this. It turns on gill function for fresh water, right? Prolactin leads to gills stopping to secrete salt and starting the proton pump. So prolactin leads to gills losing their chloride cells. They turn on this other pump called the proton pump. They turn off their sodium potassium ATPase at the gill anyway, because a lot of cells are still using their sodium potassium ATPase. But the sodium potassium ATPase gets turned off at the gill. And now they start making a lot more urine. And they're faced with this totally other thing. The proton pump allows them, now they have to conserve salts. All right. So that's how some animals have to do something different than we do with our kidney. Okay. Our kidney is mostly facing the same threat every day, the same challenges. But some animals go through a change of life where they go from something that's totally fresh to totally salty. Their kidney almost has to be turned around. I mean, I'm using, that's a slang term, but it's like the activity or the function of the kidney has to be reversed. And then in salt water, they have their adult life, and then they go back to fresh water to go spawn and die. And they have to turn their kidney around again. When they're in fresh water, they are conserving salts. When they go to salt water, now they're conserving water. All right. Sam, did I click on and off? Because I got a message that my web was unstable. Can you hear me okay?
Yes, sir. You were off for a bit, but I can hear you now.
Okay. All right. So that's something to think about. Fish going in one direction and another. I want to tell you about something else that is a comparative aspect of how kidneys function. One of the things that is going on at the kidney, I keep saying that we're getting rid of waste, but I haven't talked about the waste too much. And what waste am I talking about? The wastes that we get rid of are nitrogenous wastes. And nitrogenous wastes tend to build up as byproducts of metabolism. But as it turns out, among vertebrates, there's 3 different nitrogenous wastes that different animals use, right? Some animals get rid of their nitrogen in the form of ammonia. Some animals get rid of their nitrogen in the form of urea. Some animals get rid of their nitrogen in the form of uric acid. All of these molecules allow you to secrete, excrete, urinate nitrogenous waste, but they allow for a different amount of water conservation. Okay, so in this list that I just gave you, you conserve more water in this direction. So who uses ammonia? In seawater, squid use ammonia, but for me, a more understandable animal that secretes its nitrogenous waste as ammonia is a freshwater fish, like a sac-a-lait. That's the white perch. Okay. These animals that release nitrogenous waste as ammonia are called ammoniotelic. Ammoniotelic. What does ammonia look like? It's a single nitrogen atom with 3 hydrogens. Right? That's what ammonia looks like. Man, sometimes when you catch a freshwater fish, it pees. A freshwater fish pees, a lot of what it's peeing out is ammonia. But you're moving 1 gram of nitrogen for every 500 mils. Okay, what about animals that use urea? That's us, mammals. We are called ureatelic. What does urea look like? Urea has 2 nitrogens. Remember, ammonia only had 1 nitrogen. So urea has 2 nitrogens, double bonded O here. Whenever you get rid of your nitrogen as urea, you can get 1 gram of nitrogen out for 50 mils, right? So that's 10 times more water conservation. Mammals do that. We as mammals are ureatelic animals. Now, what about animals that use uric acid? They are called uricotelic. Uricotelic animals. Uricotelic animals conserve the most water, and you can find these sometimes in like a desert situation, or an easy one to describe is seabirds.